Hello friends, welcome to Agri Medical Concepts, and today we are going to discuss an interesting topic. There is a study which shows antimicrobial resistance in soil bacteria without using antibiotics. Yes, researchers at the Indian Institute of Science, that is IISC, and Kiel University, Germany, have shown in a study that antimicrobial resistance, that is AMR, can be found in soil bacterial communities due to microbial interactions driven by a species of predatory bacteria. So, published in the journal Current Biology, which is published by Cell Press USA, the study looked at how the presence of the bacterium Myxococcus xanthus affects the number of antimicrobial resistant bacteria in soil samples. According to IISC, M. xanthus is a predatory species that is known to release antimicrobials and other molecules to kill its prey. Human driven antibiotic resistance is a major problem, but the question is, are there other aspects that we completely ignore? This was very exciting to explore. This was said by Samay Pandey, who is the assistant professor at the Department of Microbiology and Cell Biology, that is MCB, at IISC, and who is also the corresponding author of this study. Pandey and others found that the death of M. xanthus in soil bacterial communities increased the frequency of resistant isolates, that is, bacterial cells resistant to antibiotics. These were found in many different species of soil bacteria. These cells also showed the resistance to certain antibiotics even without exposure to these drugs. When faced with starvation, populations of M. xanthus die en masse in famine-like conditions, which are very common in soil environments. These bacterial cells form stress-resistant structures called fruiting bodies that are filled with spores. During the development of fruiting bodies, only a minority of cells succeed in becoming spores, whereas the majority of the bacterial cells undergo lysis or rupture and release growth inhibitory substances into the environment. So, the researchers believe that exposure to these growth inhibitory molecules is the reason behind the increased frequency of resistant isolates in the soil bacterial community. Interestingly, not all the strains of xanthus triggered enrichment of the resistance. It was the ones with higher diversity of biosynthetic clusters that seemed to drive it. Saheli Saha, who is the PhD student at MCB and who is also the co-first author of this study, said, "We identified multiple different molecules and did a very crude classification. Individually, these molecules might not do anything, but when you put them together, they suddenly do this strange thing where they can enrich other resistant isolates." The researchers found that resistance was enriched against several antibiotics, which include commonly used drugs such as tetracycline and rifampicin. Thus, this study highlights a natural mechanism of AMR enrichment process, and understanding such microbial interactions is crucial for tackling antimicrobial resistance.
If you are having any query, any confusion from any chapter or any topic, you can write it down in the comment section, or for this purpose, I have also provided my email ID in the description box. So soon I will be back with the next interesting video. Stay connected and thanks.